requirements. Remember, first of all, do you remember what heterotrophs and autotrophs are? You can't forget that old stuff.
One cell feed, one doesn't.
Right. And when we look at Enterobacteriaceae, they're heterotrophic, but we want to know what they can use for their source of carbon. Can they use glucose? They all use glucose. They're all glycolytic. Some of them can use lactose. Some of them cannot. Remember, lactose fermenters are friendly. Non-lactose fermenters are unfriendly. By friendly, we mean could be normal human flora. Unfriendly means, yeah, it's enterobacteria, but not really for humans so much. Not really normal human flora. Although some people will carry some. Okay. So, your oxygen requirements. Obligate means must have. If it's an obligate aerobe, must have oxygen. If it's an obligate anaerobe, must not have oxygen. Oxygen will kill it. It's a facultative anaerobe, can do fine with oxygen. If it doesn't have oxygen, it's got a backup plan to do anaerobic fermentation. It's not as efficient. Remember aerobic, you get 38 ATP, anaerobic you only get 2. So they're going to grow better with oxygen, but they can still survive without it. So when you look in your test tube of broth, if you have a pure culture growing in there, the pattern of growth in broth might tell you something about its oxygen requirements. If it's a facultative anaerobe, it's going to grow throughout the test tube. However, the picture in your textbook, and Kristen also pointed out, the facultative anaerobes will probably grow better in the upper portion of the broth at the top because there's more oxygen there. The ones that the lower part of the tube will grow, but maybe not as much density of bacteria down there, right? So they'll be able to grow throughout the tube. They'll just do better with more oxygen. And do you remember what an aerotolerant anaerobe is?
So you can live without the oxygen? Or
It's aerotolerant anaerobe. So it doesn't use oxygen, but it can tolerate it. So in other words, it's not going to be killed by it.
So it doesn't need a trace of it, right?
It doesn't use it, but it won't hurt it either. Then there's a thing called a microaerophile. What do you think they want? Little bit of air. Loves a little bit of air. So if you're looking at a test tube of broth, you'll see your most growth, not at the top, not at the bottom, but somewhere in between, whatever's the right level for them. Probably a little closer to the bottom if they only like a little bit of oxygen. If they're obligate anaerobes, where are you going to find them? Down in the sediment, right at the bottom. If your test tube has a pellicle across the top, what would that tell you? They like air. They're going up to the top and they're clamoring at the top. What else could a pellicle tell you? Remember, it's sticky. It's going to stick to the sides of the test tube. So those bacteria may also have a capsule. Good. Alrighty, what else we got? Okay. If your patient has some new disease, we don't know what it is, we don't know what causes it, we want to run Koch's postulates. What do we have to do before we can even run Koch's postulates? Culture it, right. You got to get the causative agent out of your patient and onto a Petri plate or in a test tube of broth. That's the hardest part of Koch's postulates, is actually getting the material to do it with. You don't know what's causing your patient's infection. You might not even know where the infection is. You might have to sample tissue from the lungs, tissue from the bowels, whatever. If you can find where the infection is in your patient, the next trick is get it out of your patient without killing it. How is it going to live outside of your patient? Can't study it if it's not alive. So, well, maybe you can study a little bit, but not enough. All right. What do you call it when your broth test tube is cloudy?
Turbidity.
Turbidity. And what does that tell you about your broth? Yeah, you've got some microbes growing in there. And also, if you review the Chapter 6 PowerPoints, you will see If it's really cloudy, you can estimate a high number of microbes. If it's just a little cloudy, you would estimate a low number of microbes. They can actually put the test tube in like a spectrophotometer or a machine of some kind that will measure how dense the bacteria are in the test tube and estimate the number of microbes in your broth. There's actually a thing, this is not on your lab quiz, so if it blows your mind, don't worry about it, but you can look it up in your textbook. It's called the McFarland standard, which is actually a visual gauge of turbidity. And when you're working with bacteria, if your test tube is just this cloudy, that's the McFarland standard. That's how much bacteria you should have in your broth. If it's less cloudy, you're not there yet. If it's too cloudy, you got too much in your broth. Like with yours, you're like, do I have enough bacteria in my water or not? Well, some will have a lot, some will have a little. You compare it to the McFarland standard. You hold your test tube literally up to this visual picture. Is it cloudier than that one, cloudier than that one? No, almost as cloudy as that. Oh, yep, it's just this cloudy. That's the McFarland standard right there. So you can estimate your numbers of bacteria with the turbidity also. And let's see. Okay, the different kinds of medium. We have liquid medium, that would be your broth in a test tube. And we have solid medium, that would be agar in a Petri plate. Why do we want agar in a Petri plate?
To feed bacteria.
Right, but why is that better than feeding them with broth?
You can observe that.
You can observe them better because you can separate them. It's greater surface area, right? And on the surface area, you can spread them apart from one another so you can see them better. If on your lab practical, I flash up a picture, and by the way, I told the other class, your lab practical is going to be all pictures in PowerPoint slides. The timer is set, it's standardized for everyone. You all get the maximum amount of time allowed. The screen will come down, PowerPoint slideshow starts. First slide comes up, picture, question number 1, question number 2. Bing! Next slide comes.
Blank, or is it multiple
You're,
choice?
it's just like a lab practical where you just fill in a short answer. So you'll have an answer sheet, 1, 2, 3, 4, 5, you know, and see the picture. Question number 1 asks you something. What's the shape of this bacterium? Okay, what disease does it cause? Whatever. You write that in the blank.
But it's
There's
only
50
the one with
questions, 5 extra credit. They're all on PowerPoint slides. Get a minute and 15 seconds per question.
You're kidding,
It's
right?
easy.
It's on the PowerPoint.
Yeah, we'll review.
Oh,
It's
God.
easy.
Only
Please
the
tell
ones
me
on the
you're gonna review
PowerPoint.
that.
It's gonna be so easy. We will review, yeah.
Are you gonna give us the 5 extra credit points before the question? Or the extra credit before? No?
Are you
They
going
come
to
at the
give us
end.
the extra credit question before the test?
Yeah.
What does that mean?
Oh, our professor would tell us what the extra credit would be, and we'd have to learn it.
Mm-hmm.
To write an essay question.
Well, no, it
They're
wasn't an
not
essay
people.
question.
Yeah.
She was
Was
different.
was she saying that you're gonna put it's all in PowerPoint form, you mean up here, or from the PowerPoints that we have, that you've
That
gone
we've
over?
previously done, right?
Or no, you're just talking about it's a PowerPoint up here.
Yeah, the
Okay.
picture's gonna be a PowerPoint
Right.
slideshow.
That's right, okay,
So
Yeah,
the
but
first
they're all-
slide's gonna have a picture and a question.
gotcha.
Right? The question's gonna be something about what's in the picture.
And those pictures are coming from those slides which you posted online?
Or any-
Some come from the internet,
Some.
some come from pictures I took in lab, they come from all different places. Your Petri plate's a little bit open there. See?
Oh.
You should be able to recognize them.
A lot of
Alrighty, next question for the lab quiz 4 that you're gonna take this weekend in addition to your exam 2.